Hi everyone, today I'm going to be talking about Bambu, which is a transcript discovery tool developed for R to be used with long-read sequencing. Now, the reason we're so interested in transcript discovery is that transcriptomics has reached a new dynamic part of its life cycle. Originally we were starting with microarrays, then we moved on to short-read sequencing, and now we're working with long-read RNA. And that provides us with so many more opportunities than we had before. But one thing that we've really clung to over this history of transcriptomics is the use of reference annotations. Now, don't get me wrong, reference annotations are very helpful. So for example, here, if we have a long read sample, we can expect to see many reads that match the full length annotations we find in our reference annotations. However, unfortunately, it's not always that simple. Sometimes we'll get fragmented reads. So for example, we might get a fragment here, which we can uniquely assign to a particular isoform, which means quantification proceeds with no problems. But sometimes there are fragments that are a bit more ambiguous and could overlap with multiple reference annotations. And this brings us to one of the key problems with reference annotations, that they usually represent an atlas of an entire species. So every isoform that species can produce. But often our samples are much more limited. They may be at a specific time point, a specific tissue. And so often most transcripts are not actually expressed. Now, the inclusion of these unexpressed isoforms are not harmless, and in fact can be quite problematic for quantification. So just including, for example, these two unexpressed annotations means that this fragment is no longer unambiguously able to be assigned to one transcript, resulting in potential quantification errors downstream. Additionally, we might encounter reads that don't map to anything in the reference annotations. So for example, here we've got some examples of reads that don't overlap at all. How are we meant to quantify these ones? One approach is including them as part of the reference annotation as new isoforms for a gene. However, that actually can create further problems. So for example, what happens if we get a bunch of products like this, which could be a full-length transcript like this, or it might be a degraded product of one of the other reads. And so if we start including everything, we're going to only make this issue I explained in problem 1 significantly worse. So to try and address these issues, we developed Bambu. Now Bambu is a 2-module workflow that does both transcript discovery and then follows that into transcript quantification. It takes aligned reads, a genome, and reference annotations, and performs transcript discovery by training on the data using the reference annotations provided. This will produce a context-specific, sample-specific annotation set, which is fed into the transcript quantification module, where we can then produce context-aware isoform abundance measurements. Now, I'm mainly going to be focusing on how transcript discovery works in Bambu. And so what we do is we group all the reads together that share exon junctions into what we call read classes. We then match these to the given reference annotations, and these act as our labels. So if we say that if a read class, all the junctions match an annotated reference, then we consider this true, and everything else we consider unannotated or false. Then we train a model to try and learn the relationship between the different features that we calculate from these read classes, and we output a transcript probability score for every single read class, whether it's annotated or non-annotated. Now, to see if our model is actually able to capture this signal behind if a read class represents a true full-length read class, or full-length transcript or not, what we did is we removed the annotations from chromosome 1 and attempted to reclassify these read classes. So can we re-annotate the transcripts that we see on chromosome 1 without the annotations using the model that we learned? And indeed, what you can see is if we compare this to just ranking the read classes by their expression levels, so their read counts, our transcript probability score greatly outperforms when looking at the area under the curve of a precision recall plot. So now that we know that our probability score works, we wanted to delve in deeper. And one thing we realized seems to be a quite common problem across transcript discovery is it has this very unintuitive relationship between the quality of your data and then the precision of your output. So generally, when you're trying to do any results, the more you sequence, the more data you produce, the better. However, in transcript discovery, often the more you sequence, so the greater your sequencing depth is, the worse your precision gets. Now, this might sound counterintuitive to some people, but the reason is that generally most tools will use a read count threshold And so as you increase your sequencing depth, you're increasing the expression of the noise, the degradation artifacts. And so those start to get incorporated. And as you can see here, that for string type 2 and talent, as we increase the sequencing depth, we observed a decrease in the precision. And this was not an exception for our original purpose of Bambu. So what we did is we started to calibrate our transcript probability score into what we call the NDR, or the novel discovery rate, which is analogous to a false discovery rate. So we use the reference annotations to do that, and what we're able to do is actually stabilize the output to give a relatively consistent precision independent of the sequencing depth of a sample, and hopefully many other potential sample variations. Another advantage of using the NDR threshold is that it creates an intuitive relationship between the threshold that a user inputs into the tool versus the output of the precision. So generally, if you use a read count threshold or any other of these thresholds, you might double the threshold, you might triple it, but that doesn't necessarily lead to an intuitive relationship between the output. Yes, you might see an increase or a decrease in sensitivity or precision, but it's not always predictable. However, with the NDR, if you increase the, in this case, decrease the NDR threshold, you get a linear increase with the precision. So for example, an NDR threshold of 0.3 will nearly always result in a precision of the output of about 70%. So we believe this will be very helpful and very intuitive for users to be able to get a better idea of what their output would be, and also when they're trying to combine different samples together. Now next, we want to see how Bambu performs compared to some other tools. So the way we did this is we took the human genome and removed a random 50% of the transcripts, and we tried to see how many we could annotate using the samples that we had. So we provided the same input to 4 tools, so Bambu, string type 2, Blair, and Talon, and then we compared this using an external tool, GFF compare. And so what we found is that so baseline would be guaranteed a sensitivity of 50%, since 50% of the annotations were present. And we saw that Bambu was able to achieve a high sensitivity or precision across the entire range of tested thresholds compared to the other tools, meaning that Bambu does seem to work very effectively for transcript discovery. Now one other thing I want to talk about, so now that we know that Bambu works, it compares favorably to some other tools, is how do you use it? How do you decide on what NDR threshold is perfect for your sample? Now, just like every other threshold, it's always a trade-off between sensitivity and precision. But the NDR adds an extra layer of complexity, because you don't always know how many novel transcripts are found in your sample. If you did already, then maybe you don't even need to be performing transcript discovery. So what we developed in Bambu was a way that it can actually predict how many transcripts might be missing, and then by default, it will suggest to you a novel discovery rate for your sample that essentially matches it up as if you had high quality annotations like we have in human. So the way we tested this is we removed random different proportions of annotations from the human genome, so 25, 50%, and 75% in this case, and then we allowed Bambu to predict which NDR is appropriate now for that sample. So imagine it as an analog of a poorly annotated sample. And what you can see is that at the different fractions of missing annotations, it almost perfectly recommends the NDR that matches how many annotations are missing. So when we have 50% annotations that are missing, it recommends an NDR of 50% to try and capture those missing 50%. You might be wondering how this is working, and I'm happy to explain this, and maybe if you come visit my poster. But essentially what we do is we use a pre-trained model that allows us to predict which highly scored ones should potentially be annotated and which shouldn't be. So one final thing I want to talk about, I talked about the impact of quantification when we had poor transcript discovery, but also transcript discovery can improve quantification. So what we did, essentially, as before, we removed annotations from the human genome, and then we tried to detect them again and then quantify them. And so what we see is in the green dots are the transcripts that were removed and then newly identified. We were able to accurately quantify these when compared to the quantification when the full annotations were present. But most interestingly, what we found is Bambu's quantification had a greater performance, and by that I mean a lower error when looking at these missing annotations. And we believe that's mainly stemming from the fact that we do not overestimate the over annotate the genome. But what was also quite striking is that in blue, so these are the annotations that remained and weren't removed, actually also received a lower error by performing transcript discovery. So it shows that even if you're not interested in transcript discovery, doing it may still help the quantification of your sample. So with that, I just want to thank everyone. Bambu is available now on Bioconductor, and the manuscript is in revision, so it should hopefully be available to you soon. So if you're interested in any form of transcript discovery or context-aware quantification, please check us out at our GitHub. And of course, I'd like to thank everyone in my lab, especially Chen Ying, who was integral to a large proportion of Bambu. Thank you very much.